Well, I think we might make a start. Can I say I'm really glad to see such a friendly audience, because quite often I chair meetings where the audience sometimes gets kind of rough. And as you'll know, in the ME field, there's been a lot of roughness around for some time, and argument very justifiably. And you're going to hear about some really exciting stuff that's going on locally and nationally as well, and how many of the old arguments are disappearing as the scientists, not the politicians, the scientists move things forward and make some discoveries which start to ask more questions and more science is needed and so on. But I really felt this year at one of the conferences we had that the work that was being reported from people in the States and Germany and Australia, 15 different countries there, an international conference, you started to feel at last people are starting to use their brains a bit instead of just their mouths, arguing their cases and so on. And that caused a lot of trouble. People threatened each other and all sorts of things. So there's a history of ME, as many of you will know, and that has moved on quite a bit, I think, in the last year. And we'll move on again when we have that conference this year in London, and you can read about it at the back there who's speaking. People from all these countries come along, and they're all doing exciting scientific research, and young people are coming in now. They do all the research anyway. The older people just pick up the awards and things. The young people do all the exciting work, and more young people are coming in. You'll hear two speakers tonight from our friends in Norway, and the work they're doing as they start their careers. So, which one was it again, Richard? The down one? No, that one. I can do it without it anyway. Okay. You can't put it up because there's some things on. So I wanted to put that into your heads, first of all, that there is international work going on, and it is very exciting as different countries find things out and share the information. And I must say it's the friendliest scientific community I've ever worked in, having done quite a bit of work in cancer, but have seen the rivalries between different charities and groups. And this group here that Richard and Pia got on to me about 5 or 6 years ago in Norwich, after we'd had a debate in Parliament. And we still talk to people in Parliament, and we get answers to questions like, how many people in Britain have ME? Answer, a few months ago. We don't keep those figures, but we think it's about 250,000. We've also planted some other questions which are going to turn up in a book that I've just finished about ME called Science, Politics and ME. And we've got somebody, a friend of mine, asked the questions, and there's some really interesting answers from some of the medical people and some of the senior politicians there too. So we're doing all that kind of work as well to try and get things going. And the conference this year is in June, and you can find out about it there, because it really is worth coming. And what is also nice about it is patients are there alongside doctors and scientists. In the cancer world, you will never see many doctors appreciating having patients around them who come just to listen to what's going on. Because there's nothing worse than when you think you're at a conference talking about serious things, as they would put it, anyway, that you're going to bump into somebody who you're also treating and giving advice to. It seems to me that should really happen. We once set up a patients group in Parliament about cancer, and we found the doctors immediately set one up on their own, so they didn't have to come to the one and meet the patients. And I think that's a shame, because I think you should be able to talk in different languages, serious scientific language if you must, but also about some of the imminent problems that people have in their lives. And I don't just mean the illness, the pain, and not being able to get out of bed, all the basic things, muscle pains and so on. But I mean also getting your benefits. The first question at our conference this year was a medical guy from India who said, how do you get benefits in this country, for having ME? And the answer was with great, great difficulty, and it's getting worse and worse and worse as more and more people are being denied it. If you can walk to get your newspaper, it may take you 3 weeks to get it, but you're okay, you can work, and you're not getting your benefits. Some of the reasons the medics give us are just not on. They don't understand it, and they're often in the pay of the masters in the civil servants departments. So anyhow, these things are all going on and people know exactly. There are hundreds and hundreds of people who suffer some of the ailments. There's arguments about what the ailments are and so on, but I kind of always ignored that. See, I knew people were telling the truth. I always give people the benefit of the doubt first. And my first experience was here in Norwich when 3 constituents came to me at different times to say, you know, they'd had ME, you know, and they couldn't get the medical profession to admit it. Now, since I've been involved in this, that was about 8 years ago, I guess, we have been fighting to get recognition of some of the symptoms. And the Americans have got in the game now. They come to the conference and they find it easier to get private money and so on than we do, of course. And they're beginning to find things out that, you know, correlate with what we've been doing here for some time. But it is exciting when you get 2 countries. No problems with Brexit or any of that stuff. But it certainly, we get a lot of support from the NIH, National Institute of Health in the States, not for the conference itself, but for supporting research going on in labs in this country. And there's an exchange of young people between universities where they're trying to find out you know, what the causes might be. We can go on about that in the question time later on. We have some very good speakers here tonight from Norway who've come to our conference. Oystein Fluge, I think he's only come here to see Norwich City football team. It's a bad time to come, because there is a player there, a Norwegian lad plays in midfield called Teki. It's all right, you know. The fans don't shout at him too much. And Dr Ingrid Rekeland and Kari Sorland is working with Oystein. And then we've got Simon Carling, the local professor from the food research, to speak as well, and he'll tell you about the work and the ambitions that we have here. We have ambitions here. We managed, well, there was a time, I remember Richard and I about 5 years ago, and I must say Richard and Pierre are champions with Invest in ME. They do all the work themselves at running the conference, and if they got an official body to do it, it cost them about £40,000 at London rates to run a 3-day conference, and they do it all, mainly from their house, but also by getting people to come from different parts of the world and do a sterling work. And if I had any influence on people getting MBEs, not that they would take it, of course, I'd get them an MBE actually. It would be the least we could do for all the work they've done over the years, and they're just persistent. We started out getting the University of East Anglia, and I think Simon was at some of the meetings with us, which were quite savage at times, I think, Simon, to persuade the local university to take it on rather than building a private lab or anything like that. It should be in the public sector, and we got it in the end. And Simon and Tom Wildman, who works in at the UEA in the medical school, have started doing some exciting work, and you'll hear about it tonight as well. And that's good because it's local, you see. People can hear about it and support it, and go up and talk, listen to their talks, and sit in even the lecture sometimes if they'll let you in. And then not charge you, because universities are good at charging people for everything, but we would stop that. I tell you, that could be another fight. So it's very important that the public know what's going on locally, and I think there's a real opportunity with ME that that can happen now. And it gets you involved, because we wouldn't have got anywhere unless people like you support us, not just at the conference, but writing letters and asking what they could do and who they could talk to. And because it's international and bigger now, people are specialising in things. So you can send somebody to see somebody, or you can write to them or email them or what to get information. And it builds up, I think, a real community now in ME. As I say, there's not much fighting like there used to be, because people are beginning to feel there's things going on and people are starting to work together. Very basic messages, but when you work in these kind of fields, you know how hard sometimes it is with jealousies, promotions, and all these kind of things often get in the way of good science. But I think we're finding out that that's not happening. The other ambition we had, not only getting it in the local university, was having in the university a centre of excellence. That is, where there was quite a lot of people, 50 to 100 I would guess, is what I thought perhaps. And with the interest of the food research and the medical school at UEA, we have managed to fight our way into the new building that's being built up there, which will open in about a year, will it, Simon? About a year's time. So there is a prospect that we can have a real centre here and put Norwich on the map, amongst other things, in terms of good basic scientific research that makes people's lives better. because you couldn't do any of this without the patients telling us what goes on, because they have such a hard trouble against the establishment in medicine. We have sat in meetings with ministers and all sorts of people and still do it because you just have to. We know what the answer is going to be always. Well, we'll think about it, we'll see in 6 months, all that kind of rubbish you talk. But we've kept going, and I think the persistence of Pia and Richard getting on to me, getting tickets and things to get in and see people, and I've seen them in meetings, and I know their blood's boiling at times, some of the stuff they're hearing. As the medical people say, oh, there's no such thing as ME, it's just a bunch of middle-class nuts that are interested. You've heard all this kind of stuff. Well, we see it face-to-face even today, but we're beginning to wear them down, because just recently they're having to admit in the bottom line of one of their papers, there may be a biomedical factor in this, and there are people working on it, and perhaps we'll have a different attitude. But I still get invited by psychiatrists to go and see them and have a drink with them and behave. I've been able to turn that down so far. It's harder and harder, but I can tell you, they're getting really worried now. So some of you will know who I'm talking about, and the people who have been pushing that line for a long time. They're on the back foot, in my opinion, now, and I think we're going to see some exciting stuff over the next 2 years. Now, research moves at different paces. You can't promise everything, but you have a feeling. I'm sure Simon's work will reflect that, and of course Oystein's and his support will reflect that too. We're much better positioned understanding things now than we were a couple of years ago. Another couple of years, who knows, will unravel some of the mysteries. So that's my message, really, and I hope it's a positive one in the sense, here locally we're doing something to get it going. We're fighting the fight. Papers are being published in very respectable peer-reviewed journals. There's no quackery. It's absolutely spot on, the work that's going and where the publications are being put out and so on. So I'm very, very pleased to welcome you all here, and you're going to hear some really good stuff. And I think Simon's going to take over now. Simon Kari, who's at the Institute of Food Research, is going to tell you about some of the local stuff, and he'll also ask our friends from Norway to speak. So Simon, thank you.
Right. Everybody hear me okay? Is the microphone? I don't know. Okay. Thank you. So, my job is really to sort of, I guess, warm up the audience for the main act, which is Oystein and his Norwegian colleagues. I'm going to tell you about some exciting work that's been done in terms of treating ME patients. So, what I'm going to talk about is here. Just for those of you that are not familiar with ME, I'll just very, very briefly run through it. Tell you what we know, what we need to know, and why we're doing it in Norwich. And also, Ian has already alluded to some exciting new developments regarding a new building that's going up, which could be a centre for ME research in the future, well, very near future. So, why ME? So, the disease is not something that's just happened, it's not associated with, more sort of industrialised life. It goes back to probably more than 200 years. And what's interesting is that affected individuals are mainly women, and this can be from those from as young as 10 to as young as 70. And as Ian said, we don't really know how many people in the UK suffer from ME, but we believe there's at least 25,000 children and young people in the UK that have ME. And it's a long-term, lifelong illness. Very few will make a full recovery. Best estimate is maybe 6%. And this imposes a huge economic burden on the UK, not just the NHS, but everything else. And invested in charity estimates is at least £6 billion a year. So huge economic burden because we're affecting people for that throughout their whole life. And so diagnosis, I'm sure most of you know, is a major problem. And the estimate is that the vast majority of sufferers have yet to get the diagnosis of ME. And that can take more than 5 years before you get that diagnosis. We all know the symptoms can be just as bad as all the other significant autoimmune diseases, multiple sclerosis, rheumatoid arthritis, so this is not sort of mild impairment you recover from, it is a severe disease. And at least a quarter of patients will spend some time housebound and even bedbound. And what I have on the next slide just sort of illustrates this. These are the 2 case studies. These are people in this region that are affected by ME, and have very kindly agreed to share their case history with you. So this first individual has been ill for about the last 4 years. Prior to that, she had a very active lifestyle, working in London, freelance, illustration work, sales assistant, very, very busy. And she correlates the onset of the disease with a case of tonsillitis about November 2012. Since then, she's spent almost 2 years entirely housebound, and she's become very sensitive to noise. So she can't tolerate the radio, and she can't even tolerate conversations with people because it's too painful. So she has very limited social contact, and lack of mobility restricts her from doing normal things like bathing. And she also struggles to eat, so she has 6 small meals a day, which is barely enough to sustain her, so this is exacerbating all the symptoms. However, she does still have a very positive outcome in life, and she's hopeful that she will make a full recovery in the future. So she was what we call moderately or severely affected, and then we have this individual who has severely ill with ME, and she has been ill for virtually more than half her life. She was a very bright and active student before she developed symptoms around the age of 15, and she spent the last 18 years bedbound in a darkened room. She's unable to watch TV, she's very sensitive to light, and she can't read because of concentration is severely affected, and she cannot bathe or wash herself. And she can only speak via assembly, so she mainly communicates with people via text messages. So clearly severely affected. And she struggles to drink fluids, so as you can imagine, her diet is really not unable to sustain her. And the treat is that she can nibble on a piece of cheese. But again, this patient has a very positive outcome in life, and is really hopeful that she'll make some recovery in the future. So these 2 examples just illustrate the magnitude and severity of the disorder that we're trying to unravel and discover why these individuals suffer this disease. So this is what we believe ME needs: speedy and accurate diagnosis, knowing more about why it happens and how it happens, and we need treatments that treat the underlying causes and not just the symptoms, and that's very important. I think most of you are probably familiar with this, that the therapy, the mainline therapy has been cognitive behavioural therapy or graded exercise therapy. Well, that now I think we can draw a line under, because papers that came out last year, clearly the advice is it doesn't work, and we need to come up with a better form of therapy and treatment for these individuals. So what do we know about ME? Well, we're gaining some understanding, but it is still a bit of a black box for us. We think it's immune related. The symptoms seem to reflect there's something abnormal about the immune system in the patients. And a lot of the incidence or the onset of the condition is linked to an infection of some type. Viral infections are commonly associated with the symptoms. There's a high degree of incidence of autoimmunity in families which have an affected patient. That sort of implicates a genetic component, maybe a genetic predisposition. And in terms of the immune system, we know from work from Joe Cambridge's group at University College London that there are abnormal populations of B cells in these patients. And interestingly, what you'll hear later is that if we treat to eradicate these defective B cells, then this can help a large degree of patients. So the evidence is pointing to an autoimmune disorder. And what's interesting from the perspective of the research we're doing is that most people with this condition do have gut-associated disorders which may or may not be directly related to the disease. And irritable bowel syndrome is one of the most common gut-associated disorders. So that's basically what we know. So what do we need to know, or what do we need to do in order to redress that? Well, we need, as Ian said, we need a commitment to funding basic research as well as clinical research. And we need to build teams of people, because rather than having one person doing this, we need an effective team to really address and push new advances. And that means we have to work with the very best people wherever they are across the world. And again, I'll just talk a little bit about, at the end, how we're trying to bring people together across Europe and America and Australia to try and coordinate research globally on ME. And then the science hopefully will help aid diagnosis, provide better preventions and solutions, and importantly, inform people that need to know, that make decisions about healthcare and policy and where the money goes for research. So all of those things are. So as Ian said, Norwich's involvement in ME goes back only to as early as 2013, when we were approached, this is Tom Wyland and myself, to undertake an ME research program. And the charity, through crowdfunding and patient sponsorship, we were able to fund a PhD student who started in 2013. Last year in October, we recruited 3 additional students that were co-funded by the charity and the UEA, and also a student who was self-funded. So we now are building a team of young people to work specifically on projects related to ME. So just in 3 years, you can see we've gone from nothing, 1 student, to 4 students. And, you know, we're hoping this is really just stepping stones to even bigger activities. And we're also trying to recruit medical students because we're interested in educating the next generation of general practitioners, consultants in hospital, because they learn virtually nothing during the medical education about ME. So we're trying slowly but surely to change that by getting medical students involved in our research program. And again, the charity has been pivotal in helping medical students to take time out to do a research project in Norwich. And so far we've had 3 medical students, one of who's actually been in the US to learn about the ME research at Cornell University. And so we're starting to get more and more people interested and involved in the research. And as Ian said, we're working to establish Norwich as a centre of excellence for ME research. So this is what we're sort of focusing on. I'm sure some of you have seen this poster before. Is the gut the source and the cause of ME? And this is the sort of hypothesis we're interested in testing, that patients with ME have abnormal gut function and they have a leaky gut, which allows things normally kept inside the gut to get into the body to stimulate this aggressive and chronic immune response. And therapy, you know, we're starting to think about how we might be able to mirror the clinical trials that have been going in Oslo, which have been proven in Bergen, which have proven to be very effective. You know, and the team from Norway are here to help us to develop and plan for a similar trial. And then there's also some activity at Epsom St Helena where we're interested in changing gut microbes to improve health as well. So, one slide should remind you that your gut is like a long inner tube. It's about 9 meters long from your mouth to your anus, but it's not empty, it's actually jam-packed full of these things, different types of microbes, mainly bacteria, and they're there to help process and digest your food. But, like all things in life, they can work against us for reasons we don't really understand, but increasingly they're being linked to all sorts of diseases because of the changes in their makeup or changes in their activity. And so this is called dysbiosis, when something happens to change the behaviour or the population of microbes that live in your gut. And these are some of the things that we believe can instigate these changes. So diet, stress, drugs, in particular antibiotics, toxins that may be in the food, and of course infections that we may acquire through contaminated food or contaminated water. All of these can cause changes in these gut microbes, and instead of becoming beneficial, they can switch to become potentially harmful. And if our gut barrier gets damaged due to all of these stresses, then some of these microbes can gain entry into the body and stimulate this inflammatory response, which we believe can lead to all sorts of diseases. Here, not all of them are just focused on the gut, like inflammatory bowel disease, diabetes, obesity, cardiovascular disease, and interestingly, diseases that affect the brain. So Parkinson's disease, Alzheimer's disease, and we believe ME could also be another one of the diseases that are caused by events that happen in the gut. And so we're also interested in not just the bacteria that live in the gut, but the viruses. Now, there are numbers of bacteria in the gut outnumber the cells in our body by about 2 or 3 to 1. But then the number of viruses outnumber the number of bacteria by 5-fold. So by far the most popular organism in our gut are viruses. And so we're interested in knowing more about the viruses in the gut. And this is called the virome, the collective population of viruses called virome. And so why are they important? What is it? Why are they important? How can it affect disease? And is it relevant to ME? Those are some of the questions that we're now starting to address. So this just shows you a picture of, this is a single bacterial cell, and these things here are viruses. So every bacteria could be interacting with, or actually being invaded by, lots and lots of viruses. So this gives you some idea of the scale. So you think you've got a lot of bacteria, you've got even more viruses. And these are some of the types of viruses. And because we're scientists, we like to give them sort of illogical, exotic names. So we've got microviralis that look like sort of landmines. And we've got these things that look like alien ships, and these are called cordioviralis. So we've got a head and a tail. Okay, so those are what we know. And actually these here look very much like the cordioviralis. So these are the prominent families of viruses that we can look at. And they are very good at invading bacteria. So they don't invade our cells, they invade the bacteria that live in our gut. So it's like fleas on the back of fleas, if you like. And they can change the behaviour of the bacteria in our gut in several ways, and it's shown here. So what they can do is quite clever, is just not actually invade the bacteria, so they'll transfer their genetic material into the bacteria so it becomes part of the bacteria's genome. So it doesn't actually kill it. This is sort of integration here. This is called lysogeny. That's the sort of scientific name. And what's interesting is that the virus can introduce its genetic material in response to what we eat, the drugs we take, and the nutrients that are available in the gut. So there are certain things in the diet that will stimulate these bacteria to transfer their DNA into the bacteria. And usually that results in changing the behaviour of the bacteria. However, something that's far more dangerous is that these viruses can actually kill the bacteria, so they can wipe out whole populations of bacteria in the gut. This is called lytic infection, and of course this results in lots of dead bacteria, lots of bacterial products floating around, and these are very good at stimulating the immune system, and that can cause inflammation. And then we have gene transfer, as I say, so they can selectively transfer bits of their genetic material, not their whole genome, but bits of it. And this can confer antibiotic resistance on the bacteria, so the antibiotics we consume, these bacteria now become resistant to it because they've acquired the resistance gene from the viruses they interact with. And they can also transfer toxins as well. So viruses are very clever. They can change the behaviour of the cells they invade, and this is bacteria, in subtle ways. And this could underlie the changes in bacteria that we're seeing associated with lots of diseases. It's not the bacteria, it's the viruses that are causing those changes. And the sort of paradigm for this is inflammatory bowel disease. This is a very recent study that was published. And what they did is they looked at the viruses present in inflammatory bowel disease patients. So these are patients with ulcerative colitis or Crohn's disease. And what they showed that there are populations of these bacterial viruses, which we call phages, which increase in these patients with the disease. And as a consequence of this, the number of types of bacteria decreases. So this implies viruses are growing, they're killing bacteria, so the virus numbers increase, bacteria numbers decrease. And because we know these bacteria are linked to triggering disease in inflammatory bowel disease, this could actually be part of the mechanism of causing disease. And so what Maureen Hansen done, who's worked with us in Norwich, is she's already shown that gut bacteria diversity also changes in patients with mild moderate ME. So you could speculate that maybe what we're seeing in ME patients is attributable to the viruses. So this sort of led us to think, well, we need to undertake a virus study in ME patients. And this really comes about through lots of activity in Norwich, and so these are the people that have been involved in this. So myself and Tom, we just basically get out of the way and let the students and everybody else do the work. And we have Amalak Banzel, who's a consultant at St Helier Epsom Hospital, who helps us recruit our patients. We work with Maureen Hansen because we can compare our patients to American patients. And then we have people that help us do all the isolation and processing of the samples. And these are two of the medical students that have been involved in this project as well. And this is basically the process. So we collect a stool sample, we can isolate the viruses, and we can identify the viruses by their unique genetic fingerprint from sequencing. And so that gives us a picture of the virome. And if somebody may be interested in knowing a little bit more why viruses and what significance of viruses, this is a very useful review that was written by, again, 2 of our medical students. It was published late last year, and it's a very good overview. So what we've seen, so all of this analysis allowed us to identify what's present in a group of mild, moderate patients. This is about 40 patients, and the pattern is quite consistent. So we're seeing, as you can see, 3 broad families that are abundant in ME patients, and they're shown here. This is what they look like under a microscope. You see it's more of the head and the tail version that are prominent. What's interesting is because each of these viruses infect particular types of bacteria, we can also identify what their target cells are. And so that's shown on the bottom here in terms of these are the viruses and these are the bacteria that we invade. So we're getting a picture not just of viruses but also the bacteria. And this allows us to identify a sort of, if you like, a picture of what's present in an ME patient. So these are mild and moderate patients, and we're sort of developing this further now by saying, well, does this stay the same, or does it develop and change as the disease progresses? So we are attempting to characterize the virus of more severely affected ME patients. And these are some of the questions we're asking. So do these viruses kill the gut bacteria? And can these viruses cause and explain disease? And these are 2 new PhD students who joined us last year, and that's their PhD project is basically trying to address these questions. Then the other question is, well, if they're there, are they responsible for stimulating this immune response that can cause or be related to ME? So again, we've got 2 students that are trying to determine whether or not patients generate an immune response to these viruses that we're detecting in the patients. And could these viruses be the trigger for the inflammation that's seen in these patients? So these are projects that have just started. As you can see, we've gone from 1, now we're able to investigate a different aspect through building a research team. One interesting piece though is that I just want to mention here is that we've been looking at antibodies that we all secrete into the gut. There's a specific type of antibody called IgA, and it's secreted into the gut to bind and neutralize toxins or bacteria, so they can't invade the body, and they get washed out in the body. So we secrete grams and grams of this antibody into the gut every single day of our lives, and it's there basically to stop things getting into the body, particular bacteria and toxins. And so, as I said, secreting vast amounts. Now, in patients who have a leaky gut, things that normally the IgA would bind to and keep outside have an opportunity to get into the body. And when they get into the body, they stimulate a more aggressive immune response, and different types of IgA antibodies then get produced. And it's been shown in inflammatory bowel disease patients that far more of their gut bacteria get coated with these antibodies, and the bacteria get coated are specific to the disease. So the antibody may actually be recognizing the microbes that are stimulating the disease in inflammatory bowel disease. So we've been looking at this in ME patients, and this is the data from our inflammatory bowel disease patients, looking at what proportion of bacteria in a stool sample has these antibodies on their surface. And you can see in normal healthy individuals, it's a very small proportion. But in patients with Crohn's disease, ulcerative colitis, it increases. So what we have here, this is a very small study with a very small group of patients, but it's an interesting picture. So we've taken severely infected ME patients, we've taken stool samples from them, and we've taken stool samples from individuals that live in the same household. That's important because we want to control for other factors that can change or influence gut microbes, like diet, for example. So people living in the same household have to be exposed to the same diet, same environment, so we can sort of exclude those variables. Well, you can see there's quite a range in amongst the healthy household controls, but if we pair them up with their individual in the same house that has ME, you can see the levels generally go up. There are 1 or 2 exceptions, but there's an upward trend. So we're seeing something that has been shown to be linked to the causality of disease and inflammatory bowel disease. So although it's interesting, we need to do more analysis, we need more patients, we need to do more studies. But I think this is very encouraging because it may be providing important evidence that microbes may play a role in ME. Obviously, we want to know what are these antibodies binding to in a ME patient, what are the bacteria, and are these responsible for triggering the disease. So all these things we are attempting to investigate now. So what next? What else can we do to really enhance research that's been done in ME in Norwich? And Ian's already mentioned the Quadra Institute, the nice poster here. So this is a brand new institute, £75 million been invested in this on the Norwich Research Park. It will be open for business in about this time next year. And the building is on track, so we're expecting to be able to move into this building. And it's going to be a centre for gut, food and microbiome research for patient benefit. And we think this is an ideal place to focus ME research in Norwich. So this is the Norwich Research Park. So here we have the university, the North Norwich Hospital, the John Innes Centre. This is where IFR currently is, the Owen Institute and the Bob Champion Medical Research Building. So the new Quadra Institute will be here, right next to Bob Champion, close to the hospital and close to the Owen Institute. And this is an artist's impression, which I'm told is accurate of what it will look like inside. Then we'll have about 300 researchers. Importantly, it will also have part of the hospital in the building. This is endoscopy and also the clinical trials unit. So we'll have research labs right on top of the hospital facility. So patients will be able to come into the building and actually see what the researchers are doing in the building. And this is what the building looked like in December. As you can see, it's getting pretty close to completion. And we've been told that all the exterior will be finished by the end of February and then it's fitting in the interior. So everything is going according to plan. So that's a very exciting development. And this is what some of the things we'll be focusing on in terms of research activity, some of the topics. So you can see we'll be looking at early life, microbial colonisation, when babies first get colonised by bacteria. Can we ensure they get colonised by the right type of bacteria? Looking at digestion, glucose, flux, how microbes talk to us, communicate with cells in our body. The gut-brain axis is going to be very important to interest in how gut microbes communicate and influence behaviour, inflammation, healthy aging, and then therapies that are based around changing gut microbes. Inflammatory bowel disease is already one of the diseases we're looking at, central nervous system disease and dementia again. So I think all of this activity can map to various aspects of ME, so this is an ideal setting in which we can focus ME research in Norwich and indeed in the UK. So that's our ambition. The other one is we're trying to coordinate activity across Europe and even further afield. So we've been developing this with the help of the charity. It's called Emerge, the European ME Research Group, and the flags represent the representatives that have agreed to be part of Emerge, and you can see it's a pretty good representation already, and this is likely to increase as more researchers want to be involved. And you know, why did Emerge come about? Well, there's clearly some bottlenecks or obstacles to really developing, accelerating ME research. So we need a critical mass of people, obviously. The more of us that work on ME, the quicker we can advance. Funding, there's never enough money, particularly for ME research. There's a lack of a research strategy. You know, we all want to do our pet thing, but can we all work together on a common goal? And supporting infrastructure. Okay, so we need something around which we can base a lot of research activity. So Emerge came about to really define a research strategy to address research issues and the constraints for ME research. And this is what we're trying to do, is to develop and improve new diagnostics, treatments for ME, and all of these require a more clear understanding of the mechanism of disease, which I hope you appreciate is what we're trying to do in Norwich, is actually trying to figure out what causes this disease, so that we can inform on better therapies, more effective therapies. So it's a team effort. It's not just people in Norwich. We interact with people throughout the UK and further upstream. So University College London, Imperial College, University of Oxford, and then international collaborations. So it's a team, really is a team effort here, and everything we do is a joint activity. And these are some of the students that Invested ME has co-funded with the university or other investors. You can see 3 years ago we had one name. Now we're getting up to 8 names. So this is really improving our resource and ability to undertake research. And of course, none of this is possible without the patients who agreed to be part of our studies and to kindly volunteer samples their time to help us in our studies. So I will stop there, and I'll thank you very much for your attention. Thank you.
Thanks for that, Simon. That was very interesting, and I just have to say thank you for having us here in Norwich as well. We've had a meeting all day, sharing our experiences and working on a collaboration. It's been very interesting, very useful for us, and hopefully for the Norwich team as well. Just to introduce myself in the meantime, my name is Kari Sorland. I'm a nurse, and I work as a clinical trial coordinator in Bergen in Norway. And well, it's basically my job to try and organise and coordinate the efforts of the ME team. Right, we've been asked to share with you some of the work that we're doing in Bergen on ME.
Can you hear all right in the back? Can you speak up a little?
I'll speak up a little bit. We've split this presentation into three, so I'll give you a background on the studies that we've done previously in Bergen and why we have gotten into the ME research in the first place. And Ingrid will tell you afterwards about the clinical studies that we're doing at the moment. And Oystein will complete with telling you about more disease mechanisms and our thoughts on what the causes of ME might be, and also about a new study that we published before Christmas on metabolism and the energy metabolism in ME. There we go. Just a photo of the team here. We've got the bosses at the bottom, Oystein and Kari Sorland, Olav Mella, and at the top, a very friendly team of doctors and nurses and training consultants, scientists, lab technicians, and so on. It's quite a varied team. We've grown quite a lot the last couple of years. We also have collaborations with 5 different hospitals in Norway in connection with the clinical trials that we're doing. And we have collaborations with the universities in Bergen and in Oslo. Also, we have a series of international collaborations, but Oystein will get into them later on. We have to do the advertising. We have to mention our sponsors, which is the Public Trust first and foremost. They've been supporting us for 6 years, and we would not be able to do this research without them. Also, for the rituximab trial, we have received support from the Norwegian government through the Research Council and the Health Trust. And we've also received crowdfunding, which is very unusual in Norway, from the Me and You Foundation and the Norwegian ME Association. We also have to disclose the conflict of interest, which is that our hospital has supplied a patent for the concept of B-cell depletion in ME. During this talk, we'll be using the names ME CFS or just ME. But when we're talking about ME and our patients, we're talking about ME patients according to the Canadian criteria. All our patients have been evaluated according to the Canadian criteria and have been found to meet them. I'm not going to go through these in detail. Suffice to say that the core symptom is post-exertional malaise, feeling ill after activity. And there is also a need for several other symptoms like sleep disturbances, cognitive dysfunction, pain, and several of the autonomic or immune-related symptoms. When we use these criteria, it's estimated that about 0.1 to 0.2% of the population have ME, and so therefore not to be confused with general fatigue, which is about 1/10 of that number in the population. We've seen patients from the very mild to the very severe diseases, or the very severe cases, but mainly in our studies we are talking about from mild to severe very severe, are usually too complicated and too difficult to include in a clinical trial. So how does a Norwegian oncology department get involved in ME-CFS in the first place? This goes back to 2004, when Oystein Fluge got a patient with lymphoma who had also had ME for a long time. She was treated with chemotherapy, and surprisingly, she experienced considerable relief from her ME symptoms. This lasted for about 5 months before she relapsed. This was unexpected, of course, and it started Oystein and Olav Mella thinking and discussing how this could be. They speculated that perhaps ME could be an ongoing immunological disturbance, and that the symptom change perhaps was caused by an effect on the B cells. They knew that there were women, or they learnt after looking into this disease, that it was predominant in women who had ME, and it often came after infections. And they speculated that possibly this could be an autoimmune mechanism. In order to find answers to the question of the B cells, they needed a way to target the B cells, which was safe. And a well-known and relatively safe way of targeting the B cells from lymphomas and other autoimmune diseases is the anti-CG antibody rituximab. So 3 patients, pilot patients, were invited to try a single infusion of rituximab. And all 3 experienced a response. Their symptoms improved. Another ME patient, while this pilot series was being written up, another lymphoma patient presented at the department and was given 8 chemotherapy treatments, including rituximab. And she too experienced a response on her symptoms, and she's still now, 7 years later, in a moderate response. So now we have 4 cases, all pointing in the same direction, so it was time to do a systematical trial testing the effect of rituximab on ME patients. The KTS-1 trial was double-blind, it was randomised, it was a small trial with only 30 patients. All patients were given 2 treatments. Half the patients received rituximab and half the other half received placebo. And of course nobody, not the patients and not the doctors, knew which medicine the individual patient was receiving. 10 out of the 15 patients who received rituximab experienced a clinical response, and 2 out of 15 in the placebo group equally reported a clinical response. The primary endpoint, this is a criticism of this trial, is the primary endpoint, which was at 3 months, was negative, which means that after 3 months there was no difference between these 2 groups. And the doctors had expected there to be a difference because the first 3 pilots had all responded early. As it turned out later on, the typical response was much later than 3 months. So this figure shows the pattern of response and relapse in one of the pilot patients. Can you see the dot at all? This patient received 1 treatment of rituximab, and it took 20 minutes, 20 weeks, a 20-week length, before she suddenly had quite a marked response, and was in response for 14 weeks before she started relapsing. Now, when she was given a new treatment, exactly the same thing happened again. 20 weeks, then a marked response for 14 weeks, and a relapse again. So the logical thing to think was, so what if she was given another treatment here and another treatment to maintain the response? Could a maintenance treatment regime give a sustained response in these patients? So that is what we did. The next trial was a maintenance trial. It was an open study, so all the patients received rituximab, and they received the same two treatments, but follow up by 4 maintenance treatments for a total of 15 months. And again, around 64% had a meaningful clinical response, and the responses were considerably longer than they had been in the first study. But nearly all the patients did relapse eventually. After 3 years, 11 of the patients, of the 18 who did respond, had relapsed, and we're now down to 3 who are still in response. When we're talking about meaningful response, one thing we need to look at is the quality of life. Meaningful response does not necessarily mean that the patients recover completely, but that their quality of life is significantly improved. When we look at quality of life, one way of measuring that is the SF36 questionnaire, which we have used in our trials as well. It consists of 36 questions, all regarding the functional status and the well-being and the quality of life of the patient in relationship to health. This is a study that was done in 2011 showing the quality of life and the functional status of ME patients compared to other patients who have chronic diseases. And let's see, the healthy population here is non-basic. This is the healthy population around 50, and the different curves here show the different patient groups, representing the different domains like physical function, bodily pain, vitality, social function, and mental health. The ME patients have a great symptom burden, and they do score significantly lower than other chronic disease patients, both for physical function, bodily pain, vitality, social function, not so much on mental health, but still a low score, but on a scale with people with chronic depression. So this is our starting point. This is our patients before anything happens, before we start treating them. I apologise for the very, very small print here. The idea is just to give you a visual idea of what does it mean to have response. These are results from our maintenance trial. All patients have completed this SF36 questionnaire before they started treatment, and every 3 months after this. As you can see, the red here, the red groups are our 14 major responders, and we have a group of moderate responders, 4 of them, and then 10 who had absolutely non-response to the treatment. You see that they have very low scores to start with, the 3 different groups, especially here on vitality, social function, bodily pain. Mental health, the scores were not so low to start with, and they do not change in either of the group throughout the study. You can also see this down here is tiny little numbers representing the time points in the study. So at 0 and 3 months, there is no change at all. But the change starts happening here at 6 months, 10, 15, 20, reaching a max here. So in physical function, the patients reach what is pretty much the equivalent of the healthy population. Vitalities are very low for the first 2 time points, and then increasing to sort of quite a high standard. While our moderate responders experience some change, but not to the same extent as our major responders. So they do have a lift, and they do experience an improvement in quality of life, but without ever being able to say that they're cured or feeling healthy at any point. And then we have this group of 10 non-responders who unfortunately have no effect whatsoever. Similar, again, this is also one of our pilot patients. And again, we just added a bit more to this curve here. Again, this is the pattern of response and relapse. On top here, you can see the treatment. This is the first rituximab. After 30 weeks, you receive another, 16 weeks another, etc. And after the first treatment, it took 20 weeks, and then she had a response, relapse after 16 weeks, and so on. What is interesting here is the B cell numbers. What rituximab does is remove the B cells from the body. This is to start with before treatment, and just after a couple of days, the B cell number is down to almost 0. But it still takes 20 weeks before the response appears. And when this patient is in response, she actually has increasing B cell numbers in her blood. And even though she has another rituximab infusion, while she's in response and her B cell numbers are down to 0, she still has a relapse, and it takes another lag time before she's back to responding. So this tells us that it's not the B cells themselves that's causing the symptoms. The B cells are not the problem in themselves. It's some factor that's connected with the B cells, either produced by them or governed by them, regulated by B cells in some way. We've had speculation that this might be an antibody. We don't know this, but we are looking for what kind of factor this could be. The first 2 trials, although the results are promising, they have quite big limitations. The first trial was the first one ever to look at responses in ME patients after B-cell depletion. And the primary endpoint was negative. There was no difference after 3 months. And it was a very small study and very experimental. We might also have selected patients inadvertently from a particular group. We need to look at a bigger group in order to confirm this. The maintenance trial was open label, so there was no placebo group to compare with. Because we included some of the patients from the previous trial, it's difficult to really estimate any real response rate from this. So in order to come to any conclusions at all, we need to reproduce the results in a proper phase 3 multi-center trial, which is what we're attempting to do now. And that's what Ingrid is going to tell you more about. So that's it from me. I think we'll do questions after This one is working?
Okay, good evening everyone. My name is Ingrid Rekeland and I work at the Haukeland University Hospital in Bergen, Norway. I'm a medical doctor working in the oncology department. But for now I am in 100% in this ME project. I have the patients, including the patients in the clinical trials, but I'm also involved in the laboratory work. Now I'm going to speak about the 2 studies we have, rituximab trial and the cyclophosphamide trial. First, the rituximab trial is a multi-center, randomized, double-blinded and placebo-controlled phase 3 study with rituximab induction and maintenance treatment in ME CFS. a lot of difficult words I will try to explain a little bit later. But the objective is to verify or refute whether B-lymphocyte depletion with rituximab is associated with clinical benefit in patients with ME. So, B-cell depletion. What is B-cell depletion? It means reducing the B-cell number in the blood. Rituximab is a well-known drug used to treat lymphoma and also autoimmune diseases, and it's a monoclonal antibody aimed at the CD20 molecule on the surface of the B cells. So when rituximab binds CD20, the immune system will kill the B cell, and the B cell count will drop to low levels just within a few days. It's a reversible depletion, so the B cell will start to regenerate from 4 to 8 months after the last infusion. And the CD20 molecule is not found on the stem cell or not on the mature plasma cell, so the risk of infections is therefore quite low. So, all these difficult words. It's a multi-center study. We have 5 centers in Norway with 151 patients. And the study is randomized. It means it's central randomization. The patients are divided randomly into rituximab or placebo group. And the pharmacy knows the randomization code, but neither patients nor doctor knows. That means it's double-blinded. And the code is revealed in end September this year. It's placebo-controlled. That means we have the control group gets placebo solution, and it's saline added albumin. This is the reason why we added the albumin is because it's supposed to look the exact same in the infusion set. So a trained nurse will not know the difference. And we have this external monitor system for our own quality control. Sorry. So, just a short, brief explanation of the phases of clinical trials. The first 2 rituximab trials were phase 2. The first one because it was small, and the second one because it was an open study. As you can see, it fits in here. But now it's the next level. The rituximab study is larger and randomized, and therefore defined as a phase 3 study. This one is not good, but anyway. We included 151 patients, all fulfilling the Canadian criteria. The age was 18 to 66 years old, and the symptom duration was 2 to 15 years. The mild degree of symptoms, they were supposed to have a duration of the disease of a minimum of 5 years. The very severe, not included, because they are supposed to be able to come to the hospital. And the patient should not have been previously exposed to rituximab, have no primary mental illness, and no other pathology which might explain the symptoms. The treatment schedule is rituximab or placebo, induction therapy with 2 infusions with 14 days in between, and then it's the maintenance treatments are 3, 6, 9, and 12 months. The primary endpoints in the rituximab trial are self-reported forms. We did a detailed evaluation of the disease history before start of the treatment and a detailed registration of the daily functioning. This is very important because we always compare to the baseline for the entire follow-up. So we use this journal notes a lot. So the primary endpoints, as I said, it's a self-reported form with the symptoms change every 14 weeks. So it's very easy, every 14 days, sorry. So it's very easy to forget how it was in the beginning and start comparing with 2 weeks ago. So we calculate the change in the fatigue score during follow-up from the forms, and analyze the difference in course of fatigue score in the rituximab and placebo group throughout this 2 years observation, and also the number of patients in clinical response according to the predefined criteria in both rituximab and the placebo group. So for the secondary endpoints, we use different questionnaires to measure quality of life, as SF36 explained by Kari, and we also look at the patient's function level from 0 to 100% of healthy conditioning. And we have also some objective measure with the sensor armband, which is an activity armband they use for 7 consecutive days at baseline and repeated after 18 months. From this one we get steps and different activity measures. Also the sub-studies are second endpoints assessed at baseline and at 18 months. And we also evaluate the toxicity, the side effects, thoroughly. So this is just an example of what we can get out of this self-reported schemes. The 3 means that it's no change from baseline. And this is an example of a non-responder. As you can see, the total function is the same for the entire follow-up. It's a little bit less than 20% out of 100 normal function. And then you can see the fatigue score is at around 3. So this is a patient with a moderate response. You can see in the beginning, the total function level doesn't happen anything for quite a long time, until more than 44 weeks. Then it starts happening something. And then at the same time, you can see this fatigue score. 3 was no change. This is slightly changed, moderate, and major. So at this point, it starts increasing. And then she has the tool and falls down. But anyway, she comes from approximately 10% to 40%, which is a truly meaningful improvement for this patient's quality of life. This is an example of a major responder. We also have a few of these, maybe too good to be true. It starts very low, around 8% functional level, and then after 18 weeks starts improving and ends up at 65%. And what is quite interesting is that around here she feels she has a major response, but still you can see she's only 20%. So this from 8% to 20% is really, really important for the patient. As said earlier, we have focus on toxicity. We have the common side effects, which include infusion-related reactions, usually only benign reactions. And we have the infections, usually also benign infections. But there is one thing we are a little bit concerned about, and that is the late onset neutropenia. It happens when the B cells regenerate from the bone marrow. At that time, you might get low levels of the other immune cells. And at this point, if you have an infection, you need the antibiotics. So at this point, you need to be hospitalized to IV antibiotics. And other common side effects are headache, gastrointestinal symptoms. And the very rare side effects include reactivation of chronic infections, hepatitis, tuberculosis, etc. We, of course, try to exclude this in the beginning. And it's important that rituximab is given in hospital because of the anaphylaxia risk. Sorry. And you can also have, in long-lasting maintenance treatment, you can have the hypogammaglobulinemia. And what we have seen in earlier studies is that 28% get a transient worsening of ME symptoms, which is quite important now in the rituximab trial. So we do not support treatment outside clinical studies until there is more scientific evidence. So this is the 3 different sub-studies we have done. We had the CPET, the 2-day cycling tests. On day 1, we want to provoke the PEM, and day 2, to measure the effect of PEM. We do this at baseline after 8 months approximately, and we want to see if the change from baseline in anaerobic threshold will change with the symptom change. We know from other studies that patients perform worse day 2, so we want to know if this is the same in our group of ME patients. We also look at the gastrointestinal examination, it's ultrasound and also gastroscopy. And we also know that ME patients, from other studies, that ME patients have endothelial dysfunction. So we do we look at large vessel function with ultrasound technique, this one, and we look at the small vessels with the laser Doppler technique, and we don't have the time to go into this in more detail. So, next trial. 40% of the patients in the previous studies had no effects on rituximab. So, why choose cyclophosphamide? We chose this drug because 3 patients with long-lasting ME got curative breast cancer, operated, and then treated with adjuvant chemotherapy. And they then reported improved ME symptoms after a lag period, same as with rituximab. So then 3 pilot patients were treated with 6 IV infusions with cyclophosphamide with 4 weeks interval. 2 had a response and 1 were a non-responder. But this one pilot patient, she was having ME for 17 years, bedridden the last 3 years, and walked 300 steps per 24 hours before intervention, which is nothing. And then, after the treatment, she experienced a significant response of 40% function level, which she now use on walking a lot. So she has walked over 3,100 miles in the last 2.5 years. So this drug is a quite old drug used for cancer treatments with a broad immunomodulatory effect, which affects both B cells and T cells, and it's known both used in rheumatic conditions and in the oncology wards. And what is quite special with it, it has different effects on the immune system depending on dose and route of administration. So what we use is more of a rheumatic type of dose. They have 6 infusions with 4 weeks interval and with a follow-up in 18 months. And what we want to know if cyclophosphamide treatment results in significant responses and have acceptable side effects for ME patients. So the patients for inclusion are 40 patients, 25 of them are new ME patients, and 15 patients previously treated with rituximab. Both non-responders and responders with relapse are included. They all fulfill the Canadian criteria, and symptom duration at least 2 years. But here we have no upper limit of the duration. And the really severe patients and the mild one are not included. The endpoint is the same as Rituximab, and we are also focused on the toxicity. Come back to that. The objective measures with the sensor activity armband and the endothelial function and CPAT exercise test we do at the cyclomy trial as well. So this is an important slide because the treatment period is quite hard for these patients. For some reason, the ME patients have more side effects than the cancer patients. Nearly all patients got the nausea problem and a lot of constipation problem due to the antiemetic drugs. And of course, this worsening of ME symptoms were quite important, especially with the increasing number of treatments. In the beginning, it was maybe 1 or 2 weeks after the infusion, but in the end, it was nearly until the next infusion. 8 patients received the antidote for a metabolite of cyclophosphamide. The antidote is called Mesna or Eurometaxan, due to either microscopic hematuria or bladder irritation. Or if we will do trials in the future with cyclophosphamide, we will probably give this to everyone. One thing we have been also very into, because of the patients are usually in their 30s, there is a risk of premature menopause. The risk increases for women over 35. So the patients have been informed of this before consenting. But if we continue with the future studies, we are looking into fertility preservation options for our patients as well. But in this study, at least, it was 8 patients with irregular periods, 2 of them probably premature menopause, but they were more than 43 years. So, all in all, no serious toxicity, but the feedback from patients, as I said, treatment period is quite hard. But we see that a lot of people actually respond. That's why we want to continue with it, of course. But the exact number of responders we can't tell you at this moment, because we expanded the period from 12 to 18 months to learn more about the response duration. So we have also planned about the part B, the pilot study, for the really severe and bedridden patients. But this one we need to plan a little bit better because of the toxicity, and be sure that they are taken care of in the treatment period. So the status right now is that Rituximab, the treatment phase was completed in September 2016, and the results will be published in 2018 after we can finally reveal the code in September 2017. So we are super excited about that, of course. The cyclomy trial is completed in June 2017, and we hope to publish in 2017 late. So you can actually follow us if you want to. Helsebergen.no, choose English, and search for Rituximab. Thank you.
Right, you'll have your chance shortly, but the man is just going to finish it all off and tell us how it's going now. The boss. Yeah, the big boss. Oystein, you okay?
Thank you. So, we'll have had a very nice day in Norwich. We have been having an interesting meeting with a group at the research park, discussing possible collaborations. And now that my co-workers have told you about the clinical studies, I was thinking that I would try to discuss with you a little bit what we are thinking about this disease. How do we think that this could be put together? I want to start with just discussing why do we think that this could be some kind of immunological disease that in a subgroup involves these B-lymphocytes that the ladies have been talking about, possibly antibodies or maybe autoantibodies, that it could be some kind of variant of an autoimmune disease perhaps, or some other slow alteration mediated by these B-lymphocytes. What are the observations that support this statement? We think that the patterns we see from responses and relapses after B-cell depletion with this lag time, maybe 4, 5, 6, 7 months from initial treatment start until the patients start to respond, that's in line with what you can see at least in some established autoimmune diseases. We know that patients, elderly ME patients, have a very slight but highly significant increased risk of lymphoma, which is what I'm working on in daily practice in the oncology ward, and especially for these low-grade, so-called marginal zone B-cell lymphomas. And we know from these lymphomas that they are known to be associated with autoimmunity and or with chronic infections. So they are lymphomas that often develops in mucosa, in the stomach, for instance, caused by the Helicobacter pylori infection. So it's some kind of immune stimulatory, immune stimulation behind that low-grade lymphoma. At least in our studies, we have a high frequency of autoimmune diseases. More than 40% in our studies have either siblings or parents or children with an autoimmune disease, which is higher than we would expect in the normal population. Can you hear me in the back? Okay. Mainly females are affected. That's typical of the immunological diseases. At least 3 out of 4 adult CFS patients are women. But it's not like that in children. They have a 1-to-1 between the genders. It's a sudden start in many patients. 70% get this after infections. And from recent research in partly overlapping conditions, such as postural orthostatic tachycardia syndrome or the complex regional pain syndrome, if you know these entities, they have been shown to have probably an autoimmune basis. So these are the arguments why we are thinking that this could perhaps be an immunological disease with a central role for B cells, at least in some patients. So one of the questions I want to discuss with you, I will try to simplify. I know that some of you are health personnel, some of you are patients or relatives, but I want to try to discuss a little bit about the biological effector systems. Why can this immune response put up this devastating clinical picture of ME/CFS? This is some principles, schemes of how it looks. If we have the time axis at this axis and these are the B cells, the red ones, we know that from rituximab treatments you have a rapid decrease in B cells, as Kari and Ingrid has told you, and then it slowly rises. It's a reversible B cell depletion, removal of the B cells. If we think that some kind of X factor that's related to the B cells, we think that when you rapidly diminish the B cell numbers, you have a slow elimination of this X factor, which we think maybe could be an antibody or some other factor that we don't know yet. When this X factor is below a certain threshold, that's the yellow line, the patients start to improve. And then you improve for some time, and when this X factor rises again beyond this yellow line, you have a relapse of symptoms. Maybe this is the case for the short and late responses. If this X factor starts at a very high level, or the slope of this decrease is not so steep, we will touch this yellow line further out to the right. So it takes longer time to get the clinical response, and you have a shorter window before the patient relapses. Maybe this could be one of the non-responders. We never touch this yellow line, the threshold for response, even though the B cells are depleted and start rising again. And these are some of the patients that have some kind of B cell reset, like taking the PC control alt deletes in the system. You have some kind of resetting of the B cell system after regeneration, and you never have a full relapse. That's how we can think about it. So, what are the systems that's creating the symptoms? We know that the patients have an endothelial dysfunction that I told you about. Maybe that could contribute to an inadequate regulation of blood flow to meet the demands of tissues. We know that patients, like Ingrid told you, they reach anaerobic threshold at a low workload, especially if you do repeated exercise. This implies an early increase in lactate upon exhaustion. That has been measured, and that's what the patient tells us. Some patients tell me they go for 3 steps in the stairs, they feel like their legs are heavy as lead. They go to the post box and back to the house, and they feel they've been running marathon. And everyone has seen a marathon crossing the goal after running for several hours. They're just devastated, they just want to rest. The brain is not functioning, they have nausea and completely exhausted. I guess that's how ME patients feel after a short walk to the post box and back to the house. And maybe they're very severe. They have these symptoms at rest. I think so. Maybe they are alive with the symptoms of a trained athlete crossing the marathon line at rest. I think so. So these explanations for the fatigue could be, is there something wrong with the energy metabolism? Is there increased lactate production? Could that be part of the mechanism for the post-exertional malaise? And then 2 studies appeared in this year, 2016. One study from Norwich was published in 2016, showing that ME patients, when they looked in the blood, had reduced levels of several of the lipids, the sphingolipids and phospholipids, and some amino acids in the ME patient compared to healthy controls. It was highly significant. And another study showed, I was telling you a little bit about this pre-carboxylic acid cycle in a moment, but they showed that levels of the components of this cycle, which is a part of the energy metabolism, was reduced in ME patients, suggesting that they have some kind of dysfunction in the energy metabolism. I'll try to explain. So, just a little sidestep telling you that we connected to the clinical trials. We have a biobank, and because we have been doing these trials, we have 200 patients included that we have standardized biobank sampling at several time points during follow-up. And we have also characterization of these patients by all the schemes and registrations that we do, the endothelial function, the sensory hormones. These are data that we can connect to the biobank when we do analysis. These are also used for cooperations. For instance, with Joe Cambridge, who is here, looking at lymphocyte subsets, immunogenetic analysis, looking for HLA genes, several international projects. But what I will tell you about the last 10-12 minutes is the metabolic profiling that we have done in Bergen, looking at metabolites in serum, trying to see could this be something that's relevant for ME patients. For those of you, the next slide will be exactly the same, but just a little bit more complicated. This is the very simplified scheme, looking at how carbohydrates, glucose, sugars are metabolized. This is a mitochondrion. Glucose is degraded in the cytosol of the cell, transported eventually in the mitochondria, where you're supposed to make energy. And for those of you that are patients or relatives, I just want to highlight the carbohydrates, the breakdown into energy, and the lactic acid. If you have too little oxygen. If you're running past the anaerobic threshold, like everyone will if you do heavy enough training or running, for instance, then you start making lactic acid. And it's a lack of oxygen, the anaerobic way to make. Then you have little energy and lactic acid. If you have oxygen, if you're a very good trainer, you can run for a long time without making lactic acid, then you metabolize this in the mitochondria and make a lot of energy with oxygen available. Note this enzyme, one of the enzymes that's regulating a crucial step in this breakdown of glucose to energy. This is the same slide, just a little bit more complicated for those of you. The breakdown of nutrients, glucoses, to a molecule called pyruvate. This molecule is transported into the mitochondria and it's converted to acetylcoenzyme A. That's an important molecule that's used to generate energy. This is an irreversible conversion from pyruvate to acetylcoenzyme A, and that one enters this TCA cycle, the energy cycle, 3-carboxylic acid cycle, and you get a lot of energy. Note that pyruvate is the molecule that can be converted to lactate if you have anaerobic conditions. And note also that other nutrients, for instance amino acids, some of the amino acids or the fatty acids can go into and substitute and be converted to acetylcoenzyme A. And there's a lot of steps in this little bit more complicated axis. It's the same figure as the previous one, just a little bit more details. We think that there is something wrong in this system in ME patients. And some of the previous studies that we are building on, for instance, from Dr. Armstrong is here also, a study in 2012 and 2015 showing that some of the amino acids, glutamine, ornithine, glutamate, phenylalanine, was reduced in ME patients, suggesting that maybe the patients use amino acids as an energy substrate to generate energy. And these two recent papers also showed some of the amino acid decreased in ME patients. So this is also the same figure that I've shown you 2 times. I'll show you, they come again on most of the slides. Glucose, the pyruvate, into the mitochondria, and TCA cycle and energy. And we had a hypothesis that there is a defect in this central energy pathway causing changes in the serum amino acid profile. Amino acids are the building blocks for proteins that we can use for energy. So we analyzed all the standard amino acids in 200 patients and 102 healthy controls. Then we excluded those that were overnight fasting so that everyone was non-fasting. Then we had 153 patients and 102 healthy controls. And we have data on body mass, that's the weight and height of patients, the disease severity, disease duration, and their physical activity, measured with the armband for 7 days. So we know how much they move, if this could be, and the quality of life. This is a little bit complicated, but this is the essence. We need to know, understand this to be able to understand our conclusion. Because we looked at how can the cells use amino acids for energy? Where does the different of these 20 amino acids enter into this central energy pathway to generate energy, to be capitalized, breaking down? And we saw that these amino acids with red, they can be converted to pyruvate. These in blue, they can be converted to acetylcoenzyme A and B generate energy. And these green ones, all these amino acids, they can be converted to the different steps in this TCA cycle. They cannot be metabolized to energy themselves, but they can increase the capacity.
We're
5 minutes.
going to finish with our past questions. Yes, can you do it?
5 minutes, okay? Yeah. That's okay.
Tough.
Yes. What we found that we saw that looked at the amino acids that can be converted to this molecule, pyruvate. They were no different in either men or women when we compare this one, our patients to controls. No difference. If we look at amino acids that can be converted to this molecule and to generate energy, they were highly significantly lower in women with CFS compared to healthy women, but not equal in men. So there was a difference. And if you look at amino acids that can be converted to these different steps in the TCA cycle, they were also significantly reduced in women, female patients compared to healthy women, but not in men. So it seems that amino acids that are dependent on this enzyme to be used for energy, they are not interesting to the female patients with ME/CFS. But amino acids that enter downstream of this enzyme, patients try to use those. That tells us that there could be something wrong at this level in the energy metabolism. Could you get that? Because the patient tried to use energy substrates that enter this system downstream of this enzyme, but not upstream. What about the man? We couldn't find this difference in man. But what we found in men was that they had significantly increased levels of an amino acid called trimethylhistidine, which is a marker for endogenous protein catabolism. So it seems that men maybe use muscle tissue, for instance, as a source of energy substrates, while women use amino acids that they can use from serum as a source of energy. That's of course simplified, but that's how it looks. I'll finish in 2 minutes. So this enzyme complex is very complicated, and it's one of the most regulated enzymes in the body. What would we expect if there is an inhibition of this enzyme, causing a problem with the conversion of pyruvate to acetylcoenzyme? You would suppose that there is a reduced supply of acetylcoenzyme A. You would increase the conversion to lactate. You would have reduced ATP levels, and you would expect that the patient start to try to use alternative sources for acetylcoenzyme A to fuel this TCA cycle. And that's what we see. We've measured the factors that regulate this enzyme, and these are enzymes that inhibit this enzyme, and they are upregulated in ME patients. So there are several clues that there could be an inhibition, maybe also other systems, but from this system in ME patients. So to sum up with a simplified version of this slide again, this breakdown of sugars, glucose to energy. We think that there could be an inhibition of this enzyme, make patients more prone to make lactic acid. And if we put it together with the rituximab story that the ladies have told about, this is the same scheme. We think that these enzymes that are inhibiting the PDH, the pyruvate dehydrogenase, they are probably caused by some kind of aberrant immune response. Something from the B cells, maybe an antibody that's inhibiting this system. So this is the system, part of the system that creates the symptoms. What is the problem is behind this. That's the immune response, probably. We think so. Maybe this could be a mechanism for the post-exertional malaise, the endothelial dysfunction, maybe also for some of the pain problems the patients have, because this axis activates pain sensors. I think I will stop with that.
Thank you very much.